mice die at about 2 years old and bowhead whales at 200 years old. And it really looks like there's a plan built into your DNA, but it's clearly negotiable with as few as 3 or 4 transcription factors. You can go from an 80-year-old cell, skin cell, or blood cell to nearly zero. I think we're one of the few species today that could save other species. In other words, build Noah's Ark and get multiple species off the planet. I'm 68 and I feel like I'm just barely getting educated enough that I'm contributing to society. And because it just takes more time to learn everything than it used to.
In this episode, remarkable geneticist, biotechnologist, and futurist George Church joins the 632 Nanometer podcast team. In 1984, he developed the first direct genomic sequencing method, which became a foundation for the later technologies used in the Human Genome Project. He is among the first people to have his whole genome sequenced, and in 2005 became the first person to publicly release his genomic data and medical records for research purposes. Currently, he is a genetics professor at Harvard and MIT and leads the synthetic biology group at Harvard's Weiss Institute. He has also founded numerous startups such as Nebula Genomics, a company that does whole genome sequencing, and most recently Colossal, a startup focusing on de-extinction, starting with the mammoth. Yes, they literally want to bring back the mammoth to the tundra.
Thank you, George, for joining us.
Oh, my pleasure.
In preparation for the event, we actually read your paper, the 1984 genomic sequencing paper, which had 10,000 citations. And it's a two-part question. What was the state of DNA sequencing when you got into the field? And then maybe if you can go through the working principle of what made your paper so significant and your contribution so significant, and perhaps how it evolved from there. That's a lot of questions in one.
Yeah, I don't want to go on and on about it, because it is ancient history, but it is interesting. When I first got interested in sequencing, I was doing crystallography, just a distantly related field, but used sequence input. And at that time, it was very hard to sequence DNA. You can sequence little pieces of RNA. At one point, there were so few of them, I could type all of them in an afternoon. And I did that. And the methods were very primitive. And then just before genomic sequencing, my advisor, Wally Gilbert, and one of our colleagues, Fred Sanger, invented 2 different ways of sequencing DNA that were quite popular. You could sequence on the order of hundreds of base pairs at a time. And fast forward to the present, and it's trillions of base pairs. And it's about 20 million times cheaper than that. In between, the genomic sequencing method was significant for making people think about sequencing directly from genomes without amplification. And then later, it was useful again because it led to our development of next generation sequencing, our help in developing next generation sequencing. Because what it did is it involved cycles of probing and imaging that is at the core of many of the new fluorescence next-gen sequencing methods. Even though the genomic sequencing paper in 1984 is not fluorescent, but it did involve cycles of probing and imaging.
From our reading of the paper, it sounded like one of the big innovations was that you were actually able to combine many different DNA sequences into basically one pot. And then you were using radioactive oligos to do multiple reads from the same gel. Is that accurate?
Yeah, that's right. The idea is you could either use naturally occurring barcodes at the ends of the molecules you're interested in, or you could artificially introduce them. And you could do the chemistry of the whole genome in one pot. So this is the idea of molecular multiplexing, very analogous to another important technology, which is telecommunications multiplexing. But by being able to have this mixture and do a whole complicated series of steps that would normally be very expensive for one sample, you could do that same number for a very large number of samples. And, we've done multiplexing up into the billions and in many different contexts. So it was another contribution, sort of the third contribution to that paper.
That's very interesting. You were spending a lot of time in the lab working on this, right? And this is back when it was very manual. What was the most laborious part of the whole process?
Yeah, I must say that didn't really pass through our heads at the time. It's only laborious looking backwards because we were just excited how much new stuff we were getting. And when you're on an exponential, it seems like at every stage in the exponential, the whole new horizons open up. But yes, the most laborious was probably pouring the gels. Later, these were replaced by capillary electrophoresis. And the whole thing was replaced by the probing and imaging cycles that could be done. Eventually, were scaled down from the filters that the genomic sequencing paper introduced to glass slides or similar kinds of flow cells. But it was still the same kind of principle.
At the time that you were working on this, was there a competing approach that looked equally promising or even more promising that ended up not making it through the decades as gene sequencing progressed?
There was no method that competed directly with the genomic sequencing method for things like in vivo footprinting and methylation status. There was really no competing method. There were competing methods for doing sequencing, typically where you would clone the DNA into a plasmid and then just focus on one little piece of DNA. But that was not a direct competition. They were accomplishing different goals. There were plenty of methods that didn't survive. 1984 happened to also be the year that we launched the Genome Project, which for a brief moment considered a lot of options before it settled down on turning the crank on one not particularly good option, a very expensive option, but at least it had the advantage that it would work. But there were about 40 different methods, ranging from things like doing electron microscopy, like a good idea that you just look at the molecule and you see what the sequence is. But from 1984 to this day, it's still not a good way of sequencing DNA. There were some that didn't look good at the time, but turned out to be very useful, like nanopore sequencing, which I was author on the first patent on nanopore sequencing. That really took from around 1987 was when I first presented the DOE meeting until more or less the last year or two that it really became useful. So to say something didn't work out, you have to make sure you keep looking at it because it might work out in the future. Like electron microscopy might work out in the future. Who knows?
I know you're very interested in longevity, and just from a fundamental level, if two people that are already halfway through their life can produce a cell that turns into a child, like a 0-year-old human, what does that mean? Does that mean that the information is still preserved in those people and somehow aging can be reversed? What's the implication of this? And what are your thoughts on the longevity space in general? And whether from a fundamental perspective, are there any limits in terms of longevity and what can be done?
Yeah, so there are even simpler ways of getting aging reversal than procreation. But procreation tells you that there is a part of your body that is protected from aging. It's not like every single cell in your germline, all the cells that result in eggs and sperm is immortal. You make billions of them, and one of them is immortal. So typically, female fetuses have millions of precursors to oocytes, and males produce millions of sperm, per day. So that's not the recipe where you can guarantee that an organism or a cell is immortal. I think a more interesting one is that you can take a skin cell or a few skin cells and reprogram them epigenetically, as to say, using transcription factors, regulatory proteins. You can make those cells look for all the world like embryo cells. In fact, in animals, you can show that they're totipotent and that they can recreate the entire animal. Almost every cell in your body might be reprogrammable to produce all the cells in your body. And to some extent, it's seemed to be genetic and epigenetic for a long time. You get, mice die at about 2 years old and bowhead whales at 200 years old. And it really looks like there's a plan built into your DNA that says at what point in your life you're going to have multiple failures, multiple organ failures, multiple systems failures. It's almost like planned obsolescence, but it's clearly negotiable, with as few as 3 or 4 transcription factors, you can go from an 80-year-old cell, skin cell, or blood cell to nearly zero.
In terms of the work that's being done, what is the most interesting work in terms of longevity?
Well, for years, it seemed like the field was populated by either people that were not reputable or people who were reputable were not getting respect. It was not a great place to be, and there was relatively little funding for it either. There was an entire NIH institute on it, but it was one of the least well-funded institutes. But now that's changed completely, partly because of the exponential technologies like better ability to sequence DNA and RNA and collect mountains of information about the differences between old cells and embryonic cells, and the differences between people who live past 110 and those that don't, the differences between bowhead whales and mice. That accumulation of hard data has now resulted in a modest number of candidate genes, which you can manipulate either with small molecule drugs or with proteins or with gene therapies. My lab focuses mainly on gene therapies because they're more precise. They're easier to deliver in a precise way as well. It isn't necessarily the best funding that wins. That was clearly shown in all these exponential curves in electronics and in reading and writing DNA. It's often some scrappy underdog, sometimes academic, sometimes commercial, that displaces a much larger and more well-funded group. So it's a little hard to predict, but the playing field is leveled. That means everybody's got a chance, which is good for all the newcomers.
It's exciting to see the number of biotech startups now and how much easier it is to start one than it was 10 years ago. You are an advisor to, I actually don't know the number of startups, but it's definitely large. What would you say to people thinking about starting a company, either in longevity or another biotech fields? What's the recipe for a successful biotech startup?
Yeah, well, I've co-founded about 40 companies and then helped out a similar number in a non-founding role, usually as a science advisor. There's no particular recipe. I mean, one of them is having highly motivated people. Sometimes the postdoctoral fellows who made the inventions, the intellectual property, are highly motivated. Also, being in these exponential fields certainly helps, just because the progress comes collectively. Having both a team that has academic origins, but also some experience in the business aspects, that's not to be underestimated at all. Most of the failures that I've seen close up had to do with the CEO with the experience wasn't appropriate for the company that's being started. Like a CEO who's never had fewer than 600 employees could have trouble with 5 employees, things like that.
In the cases where you started companies from foundational research in your lab, what were the criteria for deciding when to translate that research to a commercial endeavor? Was it usually organic, such that postdoc came forward and was like, I really want to drive this? Or is there a set list of criteria that you personally have where you're like, okay, this needs to be true before I am willing to start a company?
Well, I found that postdoctoral fellows tend to underestimate their own technologies, which is healthy because you don't want to get into a hype cycle or start squandering your equity, getting diluted out because the product was immature. So it's good that they're cautious and modest. The way you know that it's ready is certainly when you have a major publication where it's recognized during the peer review process that it is a breakthrough. That's one indicator, but not enough. Another indicator is if you've got a product that a lot of people are asking for while it's still academic. So we've had some periods where everybody wants what we have, which is purely academic and barely working. Then we realize, as soon as this is working, then we should probably spin it out. Oh,
Do you have,
so
what's
for
an
example,
one of it?
an example was synthesizing DNA. We had a method that made it about 10,000 times less expensive, which was building it on chips. And all sorts of people, both academic and commercial, were asking us to synthesize DNA for them. These are now called DNA libraries. And we realized that if we did that, we wouldn't be getting our academic jobs done. And so we spun out a company and a series of companies that now a lot of people are using these DNA libraries.
Okay. So
And next-gen sequencing and CRISPR were other examples like that, where it was clear, like with CRISPR, we made them available at cost through Adgene, and 10,000 labs ordered them within the first couple of months. So that's another indication that there's something going on there.
okay, so after you found that you have a good idea, how do you find then the right people?
Well, there are a few ways. Sometimes a postdoctoral fellow will already be incubating something that's like a project within a lab, kind of a lab within the lab. This happened. Luhan Yang was a graduate student at a postdoc in my lab. She was one of the co-inventors of CRISPR, and she was asked essentially by the community, she and I, to come up with a CRISPR solution to the xenotransplantation problem, which had been around since the 60s, transplanting organs from animals to humans. And she built up a team of about 10 people within the lab. And then they all moved out as a unit. And that had the advantage she didn't have to hire and train people and worry about whether they're compatible or not, because that was all established in the academic setting. The other component of it is the business side. Sometimes there'll be these little, so at Harvard Business School, or it might be Sloan, there'll be competitions or there'll be business school courses where the business school students and sometimes the professors will be looking for collaborators that are scientific that don't necessarily know the business. And then they'll get together for the purposes of kind of a course exercise and then realize they're halfway through, oh, this is more than a course exercise. This is the next big thing. So those are two methods.
Just adding to this startup topic, I'm also transitioning from postdoc to the startup environment. So I'm curious, in the area of genome sequencing, what are now the key directions? Is it like to increase the speed of sequencing, or is it a matter of how to make it like in situ products for in situ sequencing, or maybe something else? If you can comment on that.
Yeah, I don't think it's priced so much anymore. I mean, the $100 genome has been around basically since 2017. That's a cost rather than a price. So by the time it gets to the market, it's marked up a little bit more than that. But it's low enough now that you could give it away for free, the way that you give away, Wikipedia and Google Maps and searches are given away for free because other parts of the economy will pay for it. And there's at least a tenfold return on investment now for a human genome. So I think one thing is clearly articulating the value of having your inherited genome, your environmental exposure to pathogens and other environmental factors, how that's valuable to you, the way that a weather map is valuable to you, can save your life. Same thing, even more so for seeing your own genomic context. So that's marketing, it's education, it's making it very simple. The way that Google Maps is a lot simpler than understanding all the complicated technology behind it, the GPS satellites and the atomic clocks and all that stuff that's behind your simple left-right directions. But sometimes there's just an instant gratification component. I think it's mainly about explaining to the public why everybody is at risk till you open up the envelope and find out whether you are or not. And so it's naive to say, oh yeah, that doesn't apply to me because there's no genetic disease in my family. Well, almost all genetic diseases are a surprise.
It would be really interesting to get to the stage, like not only diagnosing whatever the problems are, but maybe even fixing them. But I don't know, how far away are we from that stage?
We've actually been able to prevent it. Preventing is even better than fixing, because fixing implies there's been some damage and maybe some psychological damage, and then you're fixing it. But since the 80s, there's been a proof that you can handle it through genetic counseling. There's a group called Dor-Yesharim that has more or less eliminated genetic diseases, maybe 90% of them, in the subset that they look for in the population that they serve. So it's a proof of concept that it works well. It's inexpensive, much less expensive than the cures, less than $100. And the cost of the cure is $2 million. So that doesn't mean we won't bring down the costs even more, but the solution exists. But this is part of the problem, is getting people to understand how far we are right now.
A parallel question is, there's been at least some discussion and some early work in protein sequencing, which at least naively you could think, okay, that's the next S-curve where there'll be a ton of progress, which is very early, but there is no protein sequencing in nature. So you can't really borrow the way that we ended up borrowing from nature to read DNA. What's your take on protein sequencing? Will it work, and what impact will it have if it does work?
Well, there's not really DNA sequencing in nature either. There are some cool nano machines that help you, but it took a great deal of creativity and trial and error to get those turned into with the modern DNA sequencing, which is now 20 million times cheaper. Protein sequencing that has been around for a while, using things from nature. So Edmond degradation sequencing and mass spec are two methods that I've done with my own hands, date back decades. They work pretty well. And you have to ask, what is it you typically want to know about proteins? And increasingly, it has to do with the previous question, which is in situ. You want to know how proteins and nucleic acids are distributed in healthy tissue and development and in pathological cases. Less and less, it's about de novo sequencing, about learning things for the very first time. It's more about how much is there, where is it, how is it affected in pathological cases, how can we do diagnostics? And that's being able to visualize it, to see it. And microscopy now is becoming something. So instead of having two colors like H&E stains, you now have an infinite number of colors. So each pixel, each voxel in a three-dimensional image can tell you what molecule is there: protein, DNA, RNA, so forth. And that can be done with increasing collections of, say, antibodies in the case of proteins. That isn't really sequencing, but it is what you really want to do with the proteins in many cases.
I see. So you're saying that the interesting information is actually accessible with amino acids, just with the antibodies.
Yeah. And just like there's so many exponential revolutions going on in biotechnology, that's the next big one, or one of the next big ones, is being able to generate antibodies to any protein, any peptide, any carbohydrate at will, the way that you can right now make nucleic acid probes for any RNA or DNA. I think that's right on the horizon. You can see that the exponentials are kicking in for high quality antibodies and other binding molecules.
Does that cover, though, post-translational modifications? And all
Absolutely. Yes, you can do post-translational modifications that way.
That's very interesting. So you actually think that most of the progress can be made with better microscopy, or even maybe what's already available and antibodies?
I think they're complementary. I think mass spectrometry, nanopores, and microscopy are complementary. But in the end, I think of mass spectrometry and nanopores as kind of feeders that feed in, make it easier to interpret what's going on microscopically. But they're complementary. They do slightly different things, and they're all progressing.
If you had unlimited resources, no financial constraints, no people constraints, what technical problem or other problem would you work on?
Yeah, it used to be when I was starting as a student, you had to choose between what was the technically most interesting question and what was societally most beneficial. But now you can really kind of combine them all into one project. That is to say, the technology is there that you could do amazing things that are technically, intellectually, and societally interesting. But to your question, how would one prioritize? I think probably, no matter how interesting it is, you should prioritize things that are existential risk first, maybe. And that would be, on an individual level, that would be aging reversal. On a societal level, that would be the existential risks are asteroids, climate change, supervolcanoes, solar flares, things that could wipe out the planet. So anything that gets us off the planet, that kind of technology would be a good priority. I'm even publishing a paper on a speculative way of getting us out of the solar system using nanogram to picogram size probes. But I think aging has the right combination of things where timing's good. It's not that far out. It's intellectually interesting. It's societally beneficial. We spend an enormous amount of money on end of life, and everybody becomes, just after they finally get trained, they retire, have to because of health issues. I'm 68 and I feel like I'm just barely getting educated enough that I'm contributing to society. And because it just takes more time to learn everything than it used to. So I think that's an important problem. And there's a lot of avenues that are becoming feasible to tackle that right now.
You mentioned sending DNA off the planet. How do you choose between DNA or embryos? Or what order of?
I didn't say DNA. I said nanogram probes. But the problem with sending embryos, human embryos certainly, is that it would take a rather large, currently not existent machine that would nurture them all the way to adulthood. But the idea of the small probe is to have some synthetic biology tool that could replicate just enough to build a communication channel, and then everything is going at the speed of light. It is an interesting challenge because it helps us get a number of tools that we want to have anyway. The ability to do the 3D printing of any kind of object, including nanoscale objects, biological objects, and so forth. We don't really have that capacity right now. The only thing that really replicates right now are biological systems, but we could make hybrid systems that might be able to have all the best features of both. Biological systems are not particularly good at radio transmission or any kind of communication over interstellar different distances, but they could be. And likewise, probes are not particularly good at replicating from available minerals to make something from scratch. And so the hybrid systems might be quite interesting. So I think that addresses your question of whether we send DNA or embryos. We send some kind of replicating entity that's radiation resistant and very flexible. It can use a variety of very simple chemicals via synthetic biology to build two-way communication devices and 3D printer type things. So this wouldn't be 3D printers the way we normally think of it. It's more like developmental biology. I'm old. I'm 75 years old and don't have an awful lot of time left. But this is something that's recently begun to eat away at me. I like James Lovelock's Gaia principle, where he presumes that everything on the planet has a role to play or operates in some symbiotic way to further the existence of life on Earth as a whole. If everything on the planet has some role to fill or some way to contribute to the whole, wondering what human life what that contribution would be. And when I ask this question to people, the answers I get are usually, we're a virus. We're just destroying the planet. So I want to hear something that's not a simple cop-out, because I think we have more potential than that. I can weigh in on that. First of all, not every species has necessarily a long-term contribution. That many of them go extinct, but maybe they had a contribution prior to that. But specifically to humans, I think we're one of the few species, probably only species today, that could save other species. In other words, build Noah's Ark and get multiple species off the planet. If we want serious panspermia to occur, humans are the best shots for the other organisms. So I'd say that's a pretty significant component of Gaia, if the hypothesis is correct.
I like that one. I was interested in considering humanity as a potential immune system from things like last year, we sent a probe into the asteroid belt to nudge an asteroid to see if it was possible to nudge an asteroid away from orbit. And the goal of that was to see if we had the potential to deflect incoming traffic that potentially could destroy the Earth. That to me seems that not only could we act as what you just described, I consider to be a reproductive system, but we could also consider it to be immune system. The other thing that I've been wondering about is if you want to look at, what we could contribute, you have to look at what we have to offer in the first place. And the other thing that really differentiates us from other species on the planet is our self-awareness. There's no other species on the planet that is so aware of not only our big picture, but the big picture around us. And I'm wondering how that could fulfill some kind of role in planetary future.
Yeah, there are some other organisms that are self-aware, but what humans at least would regard as a primitive level, I think that's a little bit chauvinistic, but humans are particularly good at thinking about the past and the future. And it's that learning lessons from the past and generalizing, abstracting, and then engineering for the future is what gives us the ability to both help other organisms reproduce and help other organisms by providing immunity and protection. Now, sometimes we do it in a very heavy-handed way. For example, we have a milestone now that 96% of the vertebrates on our entire planet are either humans or domesticated animals. That doesn't strike one as the perfect Gaia moment, but maybe there's some bottleneck we need to go through, and that this is part of some meaningful path we're taking, just like all the extinct species played their role. Maybe this has a purpose as well, but it strikes me as a little heavy-handed.
I was wondering if we could actually use that, or say that ability to be self-aware gives us that potential to look at the bigger picture, and then gives us that potential to be able to determine, as you said, using a view of the past to look at a potential view of the future, and therefore mitigate the problems that the planet faces, including the ones that we ourselves pose. And so the part that really comes down to really interesting me is this whole concept of what does this extended knowledge or growing knowledge do for us if we tended to wipe out the planet before we had an opportunity to use it. So I've been thinking that perhaps climate change is almost a final test for humanity to be able to get its crap together, to be able to be numbered among the worthwhile species on the planet.
Yeah, I think that's an interesting way of looking at it. I'm not absolutely convinced that the people that are alive today are causing the problem, but certainly the people alive today are fixing the problem. The problem was a natural. It could be that our ancestors killed off all so many of the herbivores in the Arctic that now we have a 1,400 gigaton of methanes to be released, which dwarfs the 10 gigatons of carbon dioxide that humans produce per year. Anyway, it is a grand challenge. It is something I think we can rise to. Not every person on the planet has to participate. If a few people can figure out a cost-effective, societally acceptable solution, then, as with many other things like antibiotics, transportation, telecommunication, a small number of people can provide for the rest of the world just says thank you, or maybe it doesn't even do that.
The question is sort of related to the previous questions. One of the things humans can do, which you write about a little bit in Regenesis, is becoming transhuman. So taking DNA from other organisms and combining it with the human DNA. What do you think is the minimum viable product of becoming transhuman?
So transhuman isn't like a rigorously defined roadmap recipe. It's a little vague. I think one of the better definitions I've heard is that it's something which would make us unrecognizable to our ancestors. And I think to some extent, we already have examples of transhuman, unrecognizable technologies. For example, our ancestors, or even people today that that live isolated from the rest of society would not understand neither the technology nor the motivation for technology, space travel, telecommunications, Wikipedia, all these sorts of things. They weren't necessary back then, but they seem necessary today. Vaccines, things that save lives. But I guess to your point, what would be the next thing, the next transhumanist thing that would make our offspring unrecognizable to us? It might be already the fact that we carry our own supercomputers in our hands all day. If that integration were a little bit better, if we could travel at close to the speed of light, possibly in the form of these nanogram probes, where we essentially download ourselves, not into machines necessarily, but into a different biological substrate, which then can develop through some developmental process back into us. That would be truly unrecognizable to current generation. Those are some examples.
Just to pull on that thread, so you're suggesting something like an embryo, but when it matures, it actually has the brain that you had. So rather than just relaying some part of the memory from person to person, it's actually it's recapitulating the entire person.
Right. Sometimes people think that this can be done via electronics, and it probably can be. The question is, which is easier to make an exact copy or, something with the same biology as our brains without necessarily knowing how it works versus figuring out how it works, translating it into a completely foreign language, a different substrate, which is electronics, which currently, by the way, is thousands of times less energy efficient than our own brain. So I think if we keep it in a biological context, just get much better at developmental biology and programming active living systems, that might be something that's both more miniaturizable and more natively connected to, our current systems that work quite well. There's really only one biological system that was capable of producing all the insights of Madame Curie and Albert Einstein. There's no machine that does that yet. So maybe we should build on that, at least consider building on that legacy.
You mentioned you're working on a new project that's related to sending out probes. That's the same idea?
There's an interconnected set of ideas. Part of it is to be able to record the connectome of a brain after fixation, after death. There's the second one is to figure out the connectome of the brain in a living person. Then there's how do you both read and write that? How do you, given, say, some generic connectome, how do you modify that into a specific one? How do you build up neural networks to order? So all of this is building on developmental biology. And then the nanogram probe is about how do you transfer in the minimum package something that is capable of unfurling into something that has all these capabilities that on Earth would require a large laboratory or factory.
Okay. George, I have some provocative question. My background most like in physics engineering. So far I have been working most like with dead systems, like dead materials. So for you who work mostly like with biological or living system, it is like amazing to see that there's like life in some bacteria and of course animals, human bodies. And my question is, where is life? Where does it kick in? I mean, from physics point of view, we know there's like atoms, and then after that, I don't know, atoms come into like molecules, and then DNA, RNAs appear. But at what stage the life gets into the physical systems?
Yeah, well, I mean, so far it is not reproducible. We have not yet reproduced the process of going from non-living to living. We have ways that we can do amazing programming where we use partially living things to make different kinds of living things, but nothing that goes from the non-living to living. We think we know how to do it, but we won't really know that we know how to do it until we've done it. If our goal is survival, exploring the universe, that sort of thing, ironically, we could just do that just by leveraging all the gifts that we were given by 3 billion plus years of evolution to make marvelous variations on life that help us to get the job done. But I think we will be able to recapitulate the growth. It just depends on how long it took and whether we can accelerate it. We found that we can accelerate all kinds of things. We can accelerate developmental biology by maybe 100-fold. We can accelerate evolution by millions-fold. So we might be able to accelerate the prebiotic growth from simple molecules to polymers and cells and evolving replicating systems. There's a huge advantage that biological systems have over, say, electronic systems, in that biological systems engineer at sort of the sub-nanometer range, maybe fractions of a nanometer, while it's hard to engineer even highly repetitive systems at less than resolution of 3 or 4 nanometers. And that makes a big difference. That difference between 0.2 and 4 nanometers cubed, because it's a 3-dimensional structure, allows for evolution to occur in a very low energy and small space where you just can't get enough shots on goal with a large mechanical object.
I also work in the semiconductor space like Misha, and it's very far from being able to do anything like what biology does. A single cell can easily measure insulin levels. Many cells in your body do that, but nobody can build even a decent sized device that you could wear that would do this. It's only been done for glucose. The capabilities of chips are still very far from biology, but if you think about where the merger might happen, where do you think might be some interesting areas where the combination of nanotechnology and biology might happen next?
Well, I think the merger has happened almost since the dawn of humanity, and we keep staying merged. At dawn, we were using fire and crude rotating objects, and we're still using fire and wheels. We're pretty intimately connected to our cell phone / computer. So I think that there are definitely things that are complementary. Life is really good at printing, essentially. It's a 3D printer. We have trillions of 3D printers in less than a cubic millimeter. That's going to be very hard to do with mechano-optical electronics. What the physics and chemistry part of the world do well are things like rockets. There's no bird that can fly to the moon. Also, radios. There's not yet an organism that does a good job of radio transmission or lasers, and the list goes on and on. So I think we have these complementary worlds that have been together since we became human, and it'll just get better and better. I don't think humans need to get good at adding million-digit numbers, and I don't think computers necessarily have to get good at making a trillion 3D printers per cubic millimeter either.
George, I have a question from audience, from Louis Martin. You mentioned that the current chips are 3 orders of magnitude less efficient than the living systems. Do you predict the convergence between these two systems in the medium term?
It's possible. I mean, I kind of ban the word impossible. I would note that not only is biotechnology ahead in a certain sense, where our 20-watt brain can outperform in certain senses a 200,000 watt computer. But more and more often that 200,000 watt computer is doing things like beating us at Go, chess, Jeopardy. But the things it is beating us at are things that we were never particularly good at. We were always in awe of some idiot savant who could remember stuff really well. But that isn't the part of humanity that it's good for a laugh, but the humanity that we really care about, as mentioned earlier, Madame Curie and Albert Einstein, that kind of transcendent being able to think about the past and the future. But yes, I think that computers might get to the same low energy, high density, but in a certain sense, they don't have to, because they can program molecules to do that, and molecules can program them in return. And we have a hybrid system that is co-evolving together, and it will probably go faster as a hybrid system than it will as either one of them trying to emulate the hardest parts of the other one. I think we're going to deal with where each part has its strengths.
You spent your career in biotech as it's been growing exponentially. And like you were saying, at all times, it feels like everything is new and there's a lot of progress, even though later you look back on it and it was barely working. What's your advice to young people that are entering a field like this today for how to think in a world like that? Because it's not necessarily intuitive, and how to best position yourself. What's the right mindset for being successful when you're building on top of an exponential trend?
Well, I think the good news about the exponential is you don't necessarily have to learn everything that preceded it. The playing field gets leveled. There's a whole new set of technologies. In a certain sense, it's more complicated, but in another sense, it's simpler because we understand things better. People say, well, in the Renaissance, they could master all of knowledge. Well, there wasn't much, and what there was a lot of disinformation. Now we can understand a lot and teach and learn a lot more because we actually understand what's going on. So that's the good news. The ability to set new priorities, we're also getting, I think, much better at setting priorities that are, greener, more thoughtful of other human beings, more thoughtful of Gaia and the interconnectedness of life, not just on our planet, but outside of it. So I think all of this means that we need new people that have grown up in this world. The first generation of people that have grown up completely surrounded by computers is moving into the workplace now. It's like learning a language. That complete immersion in the language is very valuable. So that's what I would say to the next generation. You are that immersive learning generation. And we also need a generation that has grown up with quantum weirdness. So it's not weird. I mean, they've actually done quantum experiments from high school, and it's not weird. It's as intuitive as Newtonian is because they've done it with real objects. That we need that generation too.
I think one general question related to this is, if you look back on the last several decades of biotech, what was the most surprising thing that you really didn't expect, or what didn't happen you expected?
Yeah, I don't feel like I'm that easily surprised, partly because I live in a dream world of multiple futures. And so one of them happens and it's, oh yeah, that's not too unusual. What didn't happen is we still have a crazy attitude towards infectious diseases where we balk at, we meaning some of us balk at vaccines and masks. We have a strange relationship with knowing our genomes. We have these dirt cheap genomes, and yet almost nobody's getting sequenced, even though it could save them, decades of quality life or misery of a child and so forth. There's just all these anachronisms that seem to me to be something we will judge as anachronisms that we just accepted as natural. So natural, we tend to think that something that's random is intrinsically better if it's than something that's planned. So if we get a disease that makes us suffer, that's somehow random and better.
Do you think there are enough openings for experimentation? Because where you're talking about where biotech overlaps in medicine, it's actually very difficult to experiment. Do you think that it should be easier for a subset of the population to experiment or something of that flavor? Because being careful also has a cost and like the lost opportunity.
I'm not advocating radical departure. We are getting better at experimenting, actually. So we've got pioneers like Esther Duflo, here how to experiment on economics, how to hearing unequal distribution of resources, randomize and turn experiment. So that's amazing. I think we figured out how to time to doing clinical trials from 1,000 years till 12 months in the COVID crisis, where we had. So I think we're getting better at doing experiments safely. So I don't think it's a matter of cutting corners. It's a matter of reducing costs and being able to do things more in parallel. So one of my companies, Manifold Bio, is working on how to test 1,000 different protein drugs simultaneously in a higher primate or even in a human. And safety technologies are improving exponentially as well. And so as we get safer, we can do these things a little bit faster. The important thing really is that a lot of things happen in parallel. It's not necessarily the speed of each individual experiment or clinical trial that matters.
Does it seem like the mRNA trials and everything that happened with COVID, does it feel like that's going to propagate to other things where actually in reality, there's a lot of epidemics going on that are just accepted in a way because they're not an infectious disease? Do you think faster and more intelligent clinical trials modeled on what happened with COVID are possible for other diseases? And is that happening actually? Do you see that happening in the near term?
I think that there are certainly huge sectors of society that haven't learned a lesson, and that there's even bigger sections of society that are fascinated and have learned lessons from this 2-year pandemic. I think it is being applied to many things, both infectious diseases that have been neglected for years, like malaria, tuberculosis, Lyme disease. Suddenly I think there's new hope for those, not just vaccines, but preventative methods and so on. We have a method that we think we can make any organism resistant to all viruses, even viruses we've never seen before. That's the sort of thing that gets a little boost whenever we have a moment of celebration that we have conquered something I think there's a lot of silver lining to this horrible cloud we just passed through.
To pull on the thread of making all organisms resistant to viruses, as far as I understand, the idea is to basically remove parts of the DNA and not use some of the redundant codings in the DNA such that the viruses can't replicate. Is that the crux of the idea? And how do you think that that's resistant to the viruses mutating?
It's not just removal, it can be swapping, but it is recoding. It's changing the genetic code, which is nearly universal all over the world. No, you could do the math, and it is extraordinarily unlikely that these viruses could mutate in the same sense it was extraordinarily unlikely that the host could mutate to a point that it's resistant to the viruses. It requires, exceptional levels of engineering to make that many changes at once without breaking other things. And it even requires a little bit of trial and error plus to get to that point. You could design a virus just like you could design a host, but I don't think it's going to happen naturally in the wild. So I think it's a tremendous gift that we've been given, this ability to design virus resistant. But keep in mind, it doesn't make us pathogen resistant, although we have pretty good antibacterials, much better set of antibacterials than we have antiviral drugs. But even those become multi-drug resistant if we're not careful. So we really need to supplement all these high technologies with good public safety practices.
Maybe for the benefit of the listeners that don't know about this idea or haven't read about it, but also for me, because I'm not sure I fully understand based on what you just said, what changes would need to be made to the human genome and cell in order to make it resistant to all existing viruses?
It's obviously easier to do it on bacteria than human cells, but they basically have a similar number of codons, that is to say, triplets of A, C, G, and T that code for the 20 amino acids. So there's 64 codons that code for 20 amino acids. The code sufficiently so that it's completely resistant to all viruses, because no virus that we know of brings all the machinery needed for making proteins from their DNA or RNA genome. So that means they depend on the host. Normally, the host and the virus co-evolve. So the host makes one little step on its chessboard, the virus takes a counter step. But here we've taken the entire host offline, done an amazing amount of engineering so that the host is not harmed, but the code is different. Virus comes in, it's broken in so many ways that the level of mutability that you would need would be so high that you kill all the working genes as well as the genes that need to be fixed. So it's really is a matter of taking advantage of something that all viruses have in common. they're wildly different in terms of the exact mechanisms they use, how they get into the cell, etc. But they all share this one thing in common, which is dependence on the host protein synthesis decoding machinery.
Was there a lot of optimism that it would work out? And what was the dynamic eventually competing with the private group out of California? That could be interesting to hear. How did it feel at the beginning? What was the outlook and how did that turn out?
Yeah, it basically started at a meeting in Alta, Utah in 1984. There was about a dozen of us in this room that were tasked with figuring out how to measure the mutation rates in populations exposed to various energy sources. That was the congressional mandate for the Department of Energy. You know, the Department of Energy was in charge of nuclear reactors and other things that could be considered mutagenic. And we concluded in the first 5 minutes of the meeting that we couldn't figure out how to do that today. But as a consolation prize, we offered the DOE a idea that we had collectively come up with at that meeting, which was the sequencing one genome. And that might lead to ways to estimate mutation rates, which indeed it has. I think we were pretty confident that it could be done, maybe a little overconfident. Part of it was reducing the expectations. There was a very disappointing tendency from the very beginning of the project to try to cut corners, say, oh, we're just going to do the human genome and nothing else. I felt that was a false economy. Oh, we're just going to do as little redundancy as possible, which again, I felt was a poor choice because the redundancy was worth every penny because it gave you a more accurate sequence. There's a corner cutting of not doing technology development because we had a technology. All we had to do was scale it up by $3 billion. You'd want to do it again and again. You'd like to sequence lots of people and compare them and lots of animals and plants and so forth. So you really want a scalable technology, and that requires a little technology development. If you invest, and as it turned out, an investment of a few $1 million would have saved $3 billion. So after a few $1 million, we developed next generation sequencing, which made it $300 to sequence human genome rather than $3 billion. So I felt that those were some of the things I was a little disappointed about the project. Technically, it went more or less according to plan. It was delivered ahead of schedule and under budget, or about right on budget, $3 billion. But it still was disappointing to some of us. Nevertheless, right after it, as I mentioned, the whole field took off. The technology developer took off for much less money, produced the technology that we're all using today. So the second part of your question had to do with private versus public. I didn't really think that was such a big deal, honestly. There was a lot of competition within the project, even before the monopoly that was producing the instruments. They made a slightly better instrument, and then they decided that they would compete with their own customers, which is not usually considered good business practice, but it's acceptable. They didn't make a ton of money on it. They alienated some of their customers, but they managed to sell some instruments to both sides, fueling both sides. This is Applied Biosystems, or ABI, as it was called. It was a monopoly back then. We don't have any monopoly now in genome sequencing, but they did have one back then, and they sort of abused their monopoly, as monopolies tend to do, but they really don't exist as a sequencing company anymore. So it was short-lived. It kept us focused on a narrow pathway where we would get a kind of genome that could not be redone, could not be inexpensive enough to make an impact in industry or in clinics. And it wasn't the kind of genome you would deliver in a clinic because it was a so-called haploid genome, meaning it was 3 billion base pairs. Well, every cell in your body, every nucleus in your body has 6 billion base pairs, half from your mother and half from your father. And it's a different technology that you need to be able to sequence that. And that's what's relevant to clinical significance of understanding the relationship between genotype and phenotype. You can't do that with a haploid genome. And so I think that was a major mistake as well. So the Genome Project, I've said this publicly before, I think was a $3 billion waste. We should have developed the technology to get a diploid genome for sub $1,000 as our first goal. But
was there a low point where you weren't sure it was going to work at all? Or was it just clear that despite this very slow method, that maybe should have waited for better technology or made better technology? Was there always a surety that it would work, or was there a low point at some point?
I don't think there was, I mean, it's possible that somebody in the project had a depressive moment, but I don't think the project as a whole did not. I think there was a great deal of confidence. And in fact, I felt the $3 billion was a gross surplus. We didn't need that amount to do one genome. I mean, we ended up doing some smaller genomes as well as part of the deal, but that wasn't the original plan. We weren't that familiar with exponentials when we started in 1984. Most biologists, even biotechnologists, were not that aware of Moore's law, even though it had been published 2 decades earlier. They didn't know that it applied to them. But as soon as we got into the Genome Project, we suddenly saw the parallels. And I think the price would have come down faster if we had put time into technology development, as is the case in electronics. But it plummeted as soon as Genome Project was over. Then we got an exponential much faster than Moore's law.
From much of what you have told us today, it seems like biotechnology is perfectly situated in Pasteur's quadrant, where you both do fundamental science and it's also immediately beneficial to society. And I was wondering, what do you think is the role of intellectual property protection in this whole process? Is it necessary to incentivize, or is it a roadblock? And maybe it's both, depending on the situation.
Yeah, I think it's an element of both. What people sometimes forget, the alternative to patents are a way of coaxing people to share recipes. The alternative is trade secret. I think trade secrets are problematic for most patents. I think they'd be particularly problematic with biology. If you have a biotechnology that by definition has to be out in the world, then you have to obfuscate it to keep it secret. There's some technologies where the public never interacts with them. Your competitor never touches them. Like you can launch a satellite without worrying about your competitor seeing it, but you can't launch a cell phone without your competitor being able to take it apart. And most biotechnologies can also be reverse engineered. So you have to obfuscate it. And I think obfuscated biotechnology is very dangerous. That's one argument in favor of patents and other intellectual property. I don't think it's really slowing things down. Academics are almost never sued for infringement. And even companies can often get away with a great deal of playing around with other companies' technology until they decide what works for them. And then they decide who they're going to license from. And so I don't think it accelerates for the most part. I could be wrong.
Just looking at what you've done so far, although you've been involved in so many companies and so much translational science and commercialization, you actually stayed in academia at Harvard. Is there a magic to academia? Is there magic to being in an academic institution? And is that still alive for people entering it today? And how do we keep it alive?
Part of the reason that I stay in academia is because I think I'm more effective at the interface. I can help train the next generation of students that wouldn't necessarily get trained in companies the same way. It's a very different experience that an undergraduate intern will get than an undergraduate and graduate student. I also stay at the interface because I think my ADD has me make multiple contributions in different fields. While if I were, let's say, CSO at a company, I'd have to focus a great deal more and might be less productive. But these are just guesses and they're personal. It depends from person to person. This varies. Is academia, what's the future of that? I think it's pretty rosy. It's not either or, it's both. Yeah, I think there's a time in your life, especially undergraduate and graduate, where you really need to be able to explore and think out of the box and not worry about the next quarterly profit margins. So yeah, I think we need an academic, something like that. It almost certainly will evolve, but I think we're going to always need something like that.
Do you think that it actually, for example, when you apply for a grant from NSF, there's a lot of focus on what will be the application, what's the impact of this versus just exploration or some kind of fundamental or creative exploration. Do you think that actually works, or is it better to do the creative exploration in academia and have this kind of ADD and work on different problems and so on, and actually fund that, and then let it grow into something that has bigger applications on its own, rather than forcing that into the plan, having a planned application? What's your take on that?
Well, as a student, you really had to choose between whether something was pure or applied. I think now it's a harmless exercise to ask, how would you apply this? If there's some harm, it would be rigidly sticking to milestones, whether they're pure milestones or applied milestones. You need to have some flexibility, at least to explain why you're changing directions. It's more a matter of how it's implemented and enforced. The exercise of asking, how might you benefit society with this, is helpful, not hurtful. I think that's another source of creativity. It helps you think. A lot of the most abstract fields of science and mathematics often had some kind of societal muse, or explaining butterfly wings or something like that, started a field that became more and more abstract and then came back and made contributions. I think it's a net positive to both think about the pure and the societal in one grant writing exercise.
Thank you, George. We also have a question which we ask all our guest speakers. Who would like to see at our next Twitter Space event? Who would make you to attend it?
Well, geez, there's a lot of talent out there. I think I could more easily prioritize technologies than people. Some of the technologies we've talked about have all kinds of pioneers that are working on the next generation of nanopores, of aging reversal, of you name it.
If you take aging reversal as one example, or nanopores, who do you think would be interesting to talk to?
I think for aging reversal, Noah Davidson, who's in San Diego working on applying gene therapies for dogs initially and then humans, he seems like he's the closest to actually putting together all the knowledge accumulated over the decades into actual things that could reverse multiple diseases of aging simultaneously, presumably by getting more of aging, both things that are spread through the blood and things that regulate the identity of the cell as old or young. That would be aging reversal. For sequencing, I think in situ is a little more interesting than nanopores. There's some researchers working on the three-dimensional structure of the cells, of tissues, RNA and proteins. Ed Boyden's group on expansion microscopy, Tung Yim's and Ting Wu's on super resolution DNA and proteins. Those might be interesting. I mean, the obvious ones that are barely worth even mentioning is the revolutions that occurred in vaccines and in gene therapies that we saw during COVID.
I think it would be definitely fascinating to have a conversation with Elon Musk, with you as well present, about what it would take to get people out onto 1,000 planets, 1,000,000 planets, especially given the calculation you did to see how many people we could send out into the universe if we just turned all of the Earth's crust and all the carbon, oxygen, sulfur, and so on into people. It's, I think 10 to the third or 10 to the fourth more people that are alive today could be produced and sent out into space.
Right.
So that would be, have you ever passed that by Elon Musk? That almost read like something he would talk about. So I'm curious if he's ever heard that idea.
No, I haven't run it past him. I spent 2 days at SpaceX a while ago. It's actually on the internet, giving a master's class in synthetic biology. So he's heard a fair number of my crazy ideas, but not that particular one. And there are ways through these nanogram probes with even a smaller budget populating a larger number of planets. I think most people are unaware that the liquid water on the Earth is about 100,000th of the liquid water in our solar system. Liquid water, frozen or rocky or molten lava water, but liquid water, there's 100,000 times more of it in our solar system than is on Earth. So that's just our solar system. And then there's a whole bunch more out there. So you can get these probes to land, which is it's either a exercise in patience and radiation resistance, or at decelerating very tiny dust particles that are going at very high speeds. I think a conversation of that sort would be quite interesting. I'd be happy to participate.
I think on that interesting question and also answer from George, I think we'll wrap up our conversation today. So let's thank George for this inspiring event. It has been a great pleasure, George, to have you and to interact with you. And let's also thank the audience for the participation and stay tuned for our upcoming events.